Well, thank you. Margaret's talk was a really good introduction to this talk, so I'll just continue on here. She talked a little bit about why are they used and exactly what are they, and we're going to talk also about some of the risks associated with that and what the dependencies are, how the field operations and management practices play into this, and production methods and conditions, and how there's a need for balancing safety with all the benefits of using these materials. So as you all probably know, there are often these kind of biological soil amendments are added to the soil prior to planting. They add these organic and inorganic nutrients to soil, and they improve a variety of different properties for the soil that are talked about. Margaret talked about a lot of those, and these are just some of the highlights of those. So what are these? What could we say are reasonable ones? They do not include biosolids, particularly for the production of fresh fruits and vegetables, and lettuce or any other kind of leafy greens. And it does not really include raw manure, and we'll go into why that is. So we're trying to get people to understand the benefit of composting. You've heard a lot from Margaret about why that's a good thing. And we're going to talk about what's going on with raw manure and why it's not advised. So, and other treatments. So we call it untreated animal byproducts. So if there's any myriad of things that come to the marketplace, but they do not have some type of a treatment that has been shown to reduce pathogens, and particularly zoonotic pathogens. Zoonotic pathogens being those which can infect animals as well as people. So we want to have a process to treat these byproducts, including manures, but including things like feather meals and other things, because all of these products that come into the processing area, they can carry these kinds of microbes on them. They do not include uncomposted or incompletely composted animal manure and or green waste. Now, there's a lot of interest in using yard waste, but yard waste, the studies have found over the years that these yard waste can be contaminated with pathogens, zoonotic pathogens. One, because of domestic animals and grass cuttings that go into that, and other can be by the wild animals. All manner of different wild animals that even in residential areas accumulate matter in the grass clippings, on tree trimmings, on leaves, and so on. So just having those collected together, in some areas, in municipal areas, there's leaves that are put into streets, and they contain a whole lot of things that accumulate even during rainstorms that run off from other places. So that includes things that can be disinfected by a process that involves heating or composting or some other type of process. Anything that is should be treated because it's known to have the opportunity for these zoonotic pathogens should be treated by a method that is known to be satisfactory for eliminating them. A non-thermal method is not going to be sufficient. So, and that's particularly true for products that would be used to amend soils that are going to be used for growing crops that will be likely be consumed raw. So any kind of a salad type product or a variety of other things that are likely not to be typically consumed without cooking. So in the USDA National Organics Program, which a number of people follow, they're certified organic, there are rules for production of manure and compost that are used on fresh produce and in other applications. So raw manure application is not allowed within 120 days of harvest, where these edible portions would likely contact the soil directly. When you think about that, that also includes not just how it is when you put the plant into the ground and it starts to grow, but the other operations that are going on during the growth periods. So if there are cultivation operations that ensue within that 120 days, and some of that cultivation can end up having soil raised up and moved around in such a way that particles of soil will get onto the produce, then that's considered contact with edible portions. And so that would need to be taken into consideration. If there is no direct contact with the soil, then the application to harvest date would be 90 days in that case. In terms of composting, this system of National Organic Program certification does not require any particular testing of the final compost for any particular pathogens. So that's not a feature of it. It is mainly thought of as a process control scenario. So in that, they talk about the same type of things that Margaret talked about, the carbon-nitrogen ratio, and they're looking for a carbon-nitrogen ratio in the beginning during the time the pile composition is mixed. So maybe wood chips plus grass clippings plus maybe some manure, some of this, some of that. They want to have the recommendation is to have the CN ratio between 25:1 and up to 40:1 at the beginning. Also, they talk about what kind of temperatures do you need to have in order to achieve a sufficient time and temperature. So when you're talking about pathogen destruction in composting, it's not just the temperature, it's the time as well. So, and it also depends on the configuration of the piles. So if you happen to have an in-vessel system, that's one in which all parts of that material are equally exposed to the treatment temperatures that you're achieving. And so if that is more uniform in that situation, they're saying, and you can show this by your temperature records, taking temperatures at different places in that mixture in the vessel, then that will be anywhere between 131 and 170 degrees Fahrenheit. If, on the other hand, you're working in a windrow situation, not all parts of that windrow can achieve the temperatures because you've got the outer portions and so on. You still have to work within this range to get the temperature up to that high, but they're going to give you 15 days to do that. And in order to do that, studies have been conducted to show that with good measured and good equipment and careful operator attention to how the equipment is used on these piles, 5 turnings should be sufficient. Now, the bottom line to understand about this is that although these are the recommended procedures, and there are years and years and just columns of data to support these types of recommendations, it doesn't mean that the material that's generated is pathogen free. What this means is that the amount of pathogens that could have come into this from, let's say, manure poultry litter, cow manure, horse manure, whatever it started from was very significantly reduced by putting it through a process such as this, where you achieve these kinds of temperatures for these periods of time, given the kind of system that it is, whether it's an in-vessel system, a windrow system, or a static aerated pile. That's what the SAP means, is the static aerated pile. So it's a pile in which it's a large pile, which has a blower blowing air in or out, depending on how you want to operate it. But it involves this time and temperature, but it doesn't result in something that you can say is pathogen free. And that has implications for subsequent uses of the material, which we'll go into a little bit later. So in trying to understand, well, what are the risks then? If it's not pathogen free, then how do we understand how to use it? What's the best recommendations we can come up with? What are the, not only the production of these and what we need to achieve to have compost or some other treatment. Somebody talked about, I think Margaret talked briefly about pelleted material. A lot of material from chickens, and particularly laying hens, is produced into pellets. And these are really popular among a number of different growers because the pellets are much easier to uniformly distribute over acreages without messing up the machinery. Sometimes when you have poultry litter from broiler operations, all the material that is in that is pretty coarse material. And when you try to put that on one of these certain kind of spreaders, it can get hung up in the equipment, and people have had some major equipment problems with that. So there's a tendency for people to like the pelleting. To pellet the material involves a different kind of process. It also involves a heating step, but there's some additional steps there, and there's some handling operations that can be a source of recontamination of material after the treatment is occurring. And I can go into that a little bit better, but if there's any questions about that. But suffice it to say that if you're ordering any kind of soil amendment from a provider, you need to know to ask some questions about how their process has been validated and what they do to calibrate their equipment and their process so that they know that they're achieving the limits of time and temperature, and that the process is working according to the way it's supposed to work. So we're concerned about what is actually able to survive in the soil. So we've done a number of different studies for that. The place in the overall assessment of risk where this happens is pretty early on. So in addition to understanding what's the level in that manure, whatever the type of product that you've purchased and used, you need to know something about, well, how's it going to behave in the soil? Because again, you can't assume that it's pathogen free. And then how does it transfer to the crops and so on? So these are the things that have to go into the model. We conducted over a period of a number of years comparison studies on E. coli survival in surface soil, as well as when the soil's been tilled in after the soil amendments have been applied. And we did this with a number of different isolates. We looked at these in different places in the East Coast. And I think some people have also followed this over here in California. And so we now have a body of data on what the survival of these is and how they can be transferred to plants. So let's kind of go over this. These are just some features about the studies and how we did them. We did use raw, untreated manure because we wanted to understand what were some of the limits on this. It's a popular thing for a number of people to do. It does impact ultimately on some potential aspects for grazing and other farm practices. So we looked at poultry litter, dairy solids, these were separated solids, horse manure, as well as compared those for each one of the sites that we conducted these studies at with unamended control soils. So we looked at surface spreading as well as tilling in. We inoculated a sterilized poultry litter extract that we prepared in the laboratory and grew our inoculum of E. coli. We had a number of different isolates that we used. We could track these because they had certain markers that we could use to follow this. We used, some cases we applied very low population densities in the inoculum process, and others we applied very high. So we're interested in how these would behave when they're tilled in immediately after application, or if they were only surface applied. So these show you for poultry litter some of the kind of curves that we got. We tend to look at this as days post inoculation. So that's what the DPI refers to. And these are the days post inoculation. So remember, we were talking about if the product that the amendment that you applied was raw manure, which in this case it was poultry litter, and it was applied on the surface of the soil in a conventional soil. In this case, we had conventional soil and organic soil. Here was the high inoculum level. Here's the low inoculum level. And the core means that was the one where it was tilled in. So you see all these different combinations. We started out with roughly around 10 to the 3. And immediately after, in almost every single case, when we applied these, there was an immediate response in the soil on the first few days after application. When we went to take samples, population increased for E. coli in these soils. But over time, you can see that it does come down. And it comes down maybe sometimes with some blips, a little bit of increase here and there. But what we were surprised about was how long they would persist. And the fact that even after they get down to a level which we'd almost not be able to detect them, sometimes there would be a spike in those populations. And we were looking to see if those spikes were related to precipitation events. And in the end, we found that we conducted a separate study where we could control the moisture and the precipitation events, and we did see that. This was with poultry litter. So now we'll go and look at what happened with dairy and horse manure. So this was the dairy here and the horse manure down here. So again, you see the same sort of thing. You see an immediate jump up in the populations right afterwards, and then it fluctuates over time. And that also seemed to correspond to soil moisture. Whether it was starting with a high inoculation or a low inoculation, they all ended up persisting to some extent for very much longer periods than what the National Organic Program limits show. But they're at low levels. We put this to our statistician and they came up with 6 different models. So there's one that's a very simple exponential loss model here. There's another one where you get this initial increase and then a very slow die off. They're just different configurations based on the data that we had from about 14 different seasons in several locations throughout the Northeast. And so then we have these others. So we then gave this data to some collaborators at the Food and Drug Administration, and as well as our own statisticians. And they did a different kind of analysis on this, which is looking at, well, of the different factors that we had, the different kinds of inoculum, the inoculum and the amendment, including the management, and whether there was depth involved in the particular samples, and whether the site that we were at, the year and the season had an effect on the outcome of the ranking of the population demise or not demise. The inoculum itself, the amendment type, and the depth of the sample, or the management practice of how it was done, season. And as you can see, there's a ranking here as to the relative importance of all of these different factors that we could glean from the data set that we had, because we had a lot of different data from all these different places, but these were the main factors that we see. And the interesting thing about this is that of the physical factors that we see is that the depth of the sample, so the tilling in, that was an important feature. Actually the inoculum itself, whether it was high or low, that was an important feature. The site and the year, and this turned out to be very much weather related. So in terms of how can we use the data that we got to predict how long these organisms, in any year where we don't already have the weather occurrence, how long would it take for those to disappear? It turns out that there's a lot of variability there. And then the season, that was it. Moisture, soil moisture in it itself, which is whether we get a rainfall event or whether there's an irrigation event, that's another factor that's really high up on the predictability range. So just as a matter of comparison, then we want to look at, well, sometimes the question comes up, well, should we just try to measure the particular organisms that we think are, likely pathogens in these products? And so we look at, well, what's actually out there in terms of the regulations. So we have the USDA National Organic Program regulations for products that would be on the market. US EPA has some regulations for types of composts that are on the market. There's some other places, the LGMA has their regulations, other places have made up their regulations. So for E. coli, NR means there's no requirement. There's no requirement in the USDA program, EPA program on some of these. LGMA had a requirement for this particular strain of E. coli. National Organic Program doesn't have anything. It's, as I said before, a process-based system. US EPA for biosolids has a process-based system. So fecal coliforms and salmonella are the two that they require. A lot of people, as you see, have this requirement for fecal coliforms. So just to clarify about fecal coliforms, that's different than total coliforms. So when you're reading along and you'll see people referring to coliforms, you might want to ask, well, which group of coliforms are you referring to? Total coliforms can be just about anything. Fecal coliforms have very special detection requirements in terms of the methodology that's used, and it distinguishes them from the all other stuff that's named coliforms, but probably doesn't necessarily have anything to do with fecal material at all. So using this term fecal coliforms, these organizations have felt that this is a pretty good indicator for a process control system. It's not an indicator for, it's an indicator of, could there possibly be some pathogens? Yes, and so their limit is 1,000, most probable number, which is their estimate per gram of sample. And they have specifications for how to do that, kind of analysis. Just so you can be aware that there is a way to measure these things in these compost or other types of materials, but nothing is easy to do this way. Getting the samples, and the U.S. Composting Council right now is going over reviewing what the adequacy of the current methodologies for actually getting the samples and processing the samples, and should they even be using some kind of a system like this for all the different kinds of products that are hitting the market. So that's being revisited as we speak. So in conclusion, I would say that foodborne illness pathogen populations from animal feces, whether it's a poultry, cow, or horse, or ducks, or sheep, or goats, if it's a fecal material, they have also been found in green waste. So there's no exception that we can easily make a case for green waste, because we know that they're there. The populations in these kinds of materials for compost and for when they get incorporated into soil, even if there should be something left in the compost, they will typically decline in about 60 days after that application method to almost undetectable. Okay, there can be some spikes after that because of precipitation or irrigation or some other very local type of things, but their very long-term type of persistence is extremely rare, but it does occur infrequently. And it's just something to be aware of so that you don't hang on to the idea that, oh, it's composted, it's pathogen-free. It is not claimed to be pathogen-free. It is claimed to be reduced substantially in pathogens. There are just multiple numbers of different kinds of factors that influence the survival of these pathogens once the materials are incorporated into soil. And the location, the site, the year, the season, the soil moisture, and a whole myriad of other things, how well the tillage is going and so on, do impact these. So with that, I think I will just conclude with this and say that we're trying to do the best we can with balancing the safety with the benefits and preventing their transfer onto fruits and vegetables. There are a number of things that impact this other than the heating and the thermal treatments that people devise. Handling and management are really big factors. And if anybody wants to talk about that, we can discuss that at a later time. So with that, I just call your attention to the two main publications that are available for your perusal to substantiate the information that I gave here. One is and this one, and the other one is this. This was done in connection with the Food and Drug Administration colleagues at College Park, Maryland. So thank you.